So, a very good evening to one and all present here. Welcome to the week 8 live class session on the course Introduction to Complex Biological Systems. I am Srijan Sen, and today I will be taking your live class session. So, yeah. So let's start with the class. So, first we will be going with the lecture in brief, and then we will be solving a few question and answers. Right? So I'm sharing my slide with you all. So I guess my screen is visible to you all. So in today's lecture, our main topic of interest is basically evolution, that is the history of life. So how did life evolve from simple single-celled organisms to the enormous diversity of organisms that we see today? Or why are humans, bacteria, and plants built from the same fundamental biological molecule? So, while we explain this, the answer lies in evolution. That's a gradual process through which organisms change over time through the genetic variation and the natural selection. So, life on Earth did not appear suddenly in its present form. Instead, it's evolved over billions of years through a series of major transitions. So, based on this short introduction or an idea about a few questions, we will go into the depth of what is evolution, the history of it, and how has life evolved. So basically, Earth was formed around 4.5 billion years ago in a highly hostile environment. So early Earth was characterized by intense volcanic activity, high temperature, frequent meteor impacts, absence of oxygen in the atmosphere. Now, despite these harsh conditions, the first living organisms appeared around 3.5 billion years ago. So the earliest life forms were quite simple. They were single-celled. They were prokaryotic. They lived in an anaerobic environment, meaning that oxygen was not present in the atmosphere. Now, there are certain examples about the early bacterial and the archaeal organisms. So, over time, the biological evolution introduced several key milestones that dramatically transformed life on Earth. So, some of them were emergence of oxygen in the atmosphere, evolution of eukaryotic cells, origin of multicellular organisms, diversification of animals during the Cambrian period, and these events collectively shape the biological diversity that we see today. One such important event is basically the Great Oxidation Event. What was that? So, there was a pivotal transformation in Earth's history around 2.4 billion years ago, which was characterized by a dramatic and a permanent increase in the atmospheric oxygen levels, are driven by the cyanobacteria that's capable of oxygenic photosynthesis, which released oxygen as a byproduct of splitting water molecules. So before this event, the Earth's atmosphere contained very little oxygen. It was mainly methane, carbon dioxide, nitrogen, water vapor. Now, the major driving force behind the oxygen increase was the particular bacteria, the cyanobacteria. So, what it does, it performs oxygenic photosynthesis, meaning that they split water molecules to generate the oxygen. So, how can we simplify the reaction as water plus carbon dioxide plus in the presence of light gives us organic molecules and liberation of oxygen. So this set the stage for the evolution of complex life. Now the oxygen that was released by the cyanobacteria initially did not accumulate in the atmosphere because it reacted with certain elements that were present in oceans, that is particularly oxygen. That's particularly oxygen reacting with iron. Now this reaction formed several iron oxides which accumulated in the different layers called the banded iron formations, right? And these geological structures are still visible today that provide evidence of the early oxygen production. So eventually with the over the years, after most oxygen reactive materials were oxidized, oxygen began to accumulate in the atmosphere. Now this transformation changed the earth from a reducing atmosphere to an oxidizing atmosphere. Okay. Now, there are certain biological consequences because of the increase in the oxygen that happened in the atmosphere. So that led to the extinction of many anaerobic organisms. So oxygen is toxic to many anaerobic microbes. So many life forms disappeared. Oxygen enabled aerobic respiration, which produced more and more ATP than the anaerobic metabolism. So that led to the evolution of anaerobic metabolism. And there was the formation of the ozone layer, which was very, very important. So oxygen in the upper atmosphere formed ozone, which protects the earth from harmful ultraviolet radiations. So this protection allowed life to expand into more diverse environments. Thus, the great oxidation event laid the foundation of the complex life. But after this great oxidation event, there was the snowball Earth hypothesis. So, Earth experienced dramatic climatic changes. One such hypothesis is the snowball Earth hypothesis. So, according to this theory, the Earth once became almost entirely covered in ice. And this process began with the oxidation of methane. Methane, as we know, was a powerful greenhouse gas in the early atmosphere. When oxygen levels increased, methane was oxidized into carbon dioxide, which is a weaker greenhouse gas. This reduced the greenhouse effect and caused global cooling. So as the temperature started to drop, ice began to spread across the planet. Ice reflects the sunlight much more efficiently than water or land or water. Now, this increased planetary albedo, meaning that more solar radiation was reflected into space. As a result, Earth cooled even further, causing more and more ice formation, which was indirectly a positive feedback loop that may have frozen the planet. But eventually, volcanic activity that was quite relevant during those times released large amounts of carbon dioxide. So what happened? Carbon dioxide started accumulating in the atmosphere because weathering processes were suppressed by the ice coverage, and this cycle had profound effects in the evolution of life. And many organisms went So the thing that I was saying was, so with the increased volcanic activity, which released large amounts of carbon dioxide, so carbon dioxide started accumulating in the atmosphere because weathering processes were suppressed by the ice coverage. So once enough CO2 accumulated, a powerful greenhouse effect melted the ice, and this cycle had profound effects on the evolution of life. So many organisms went extinct, but some survived in isolated refuges such as the ice-free ocean pockets, hydrothermal vents, and these surviving organisms later repopulated the earth. So evolution of eukaryotic cells was one of the most important evolutionary steps, where eukaryotes are the cells with a nucleus, they have membrane bound organelles, they have complex internal organization, and a widely accepted explanation is the endosymbiotic theory. So, what does this theory say? So, according to this theory, there is an ancestral prokaryotic cell engulfed another bacterial cell. So, instead of digesting it, the engulfed cell survived. Over the time, the two organisms formed a symbiotic relationship, and eventually the engulfed bacteria evolved into modern organelles. So, some examples that you can find is that mitochondria originated from aerobic bacteria. You have the chloroplast originating from photosynthetic cyanobacteria. You have certain evidences which supports this theory, like mitochondria and chloroplast have their own DNA. They divide independently. They resemble bacteria in size and structure. And this symbiotic integration was a major innovation that allowed the evolution of complex eukaryotic cells. Now, the transition to multicellularity, where comes the colonial theory, the symbiotic theory, and the syncytial theory. So, multicellular organisms evolved roughly around 1 billion years ago, and there are several of these theories that explains how multicellularity actually arose. So, when we go into the colonial theory, so this theory suggests that the multicellular organisms evolved from colonies of unicellular organisms. So the cells remained attached after division. Over the time, cells specialized for different functions, communication system evolved. So modern example is basically the chanoflagellates, which forms colonies resembling early animal ancestors. The syncytial theory speaks of, it proposes that multicellular organisms evolved from a multinucleated cell. Internal membranes later formed around the individual nuclei, creating separate cells. And the symbiotic theory. So the symbiotic theory says that different species of unicellular organisms formed cooperative relationships. Eventually, these organisms became interdependent and functioned as a single organism. And from here, you can understand that there are certain advantages of multicellularity. Like, large organisms are less vulnerable to predation, so the larger size matters. Division of labor, where different cell types formed perform specialized functions, and there is an environmental stability where the multicellular structures provide protection against environmental fluctuations. However, certain challenges of multicellularity requires communication between the cells, transport systems for nutrients and oxygen, and mechanisms for waste removal. So these challenges led to the evolution of complex tissues and organs. Now the Cambrian explosion is or occurred approximately 541 million years ago. So this was a relatively short geographical period during which most major animal groups appeared. So during this time, body plans diversified rapidly, complex ecological interactions developed, animals developed hard shells and skeletons. Now, major animal groups that appeared include the arthropods, the mollusks, the echinoderms, early vertebrates, and what is most important is that this event dramatically increased biodiversity. So, the possible causes could have been several factors. So, the probable causes includes the environmental factors like I mentioned about the increased oxygen levels. You have the shallow marine habitats, stable climates. Okay, uh, there could be multiple genetic factors like the evolution of the Hox genes, the gene duplication events, predator-prey interactions, competition for resources. So together, all these factors triggered rapid evolutionary innovation. However, there are certain challenges and controversies like the rapid evolution versus gradualism. So there is always a debate over whether the Cambrian explosion was a true rapid event or an artifact of incomplete fossil records. There could be bias in the fossil records, so preservation favors organisms with hard parts, potentially overlooking earlier soft-bodied life. That's quite possible. The Precambrian complexity, where the evidence from the Ediacaran period suggests a longer timeline for the emergence of complexities. But the controversies, which there is good to have controversies, but certain controversies can never be resolved because you cannot go back to the past and understand it. Now, the question is, why do we study evolution? What is the requirement of studying evolution? So, evolution is not studied only through fossils. So, modern scientists use molecular biology to understand evolutionary relationships. One modern method is the sequence alignment. For example, you are comparing hemoglobin and myoglobin sequences that reveals evolutionary relationships. Although these proteins have different functions, they share similar structures. So only about 17% of the residues are identical, yet the structural similarity indicates a common ancestor. So this demonstrates that proteins can diverge significantly while maintaining the similar functions. So, like I was telling about the fossils, so the fossil Lucy refers to the partial skeleton of an early hominin species that was discovered in 1974 in Hadar, Ethiopia by Donald Jonassen and his team. So you can see the structure of hemoglobin and myoglobin to be quite similar, though they have certain structure sequence differences. If you see that only 28 of the 150 residues, that 70% are identical in the two proteins, around 45% of the residues are identical because the alpha and the beta subunits of hemoglobin, and sequence identity between the two subunits of hemoglobin and myoglobin is only 30% and 26% respectively. So these are the different ancestral diversifications that have occurred starting from myoglobin to hemoglobin to erythrochronin, clam hemoglobin, worm hemoglobin, things like that. So there are multiple, they have the same ancestors. Right. Now protein 1 is more similar to which of them you can understand based on the sequences that they basically have. So you have protein 1 where you have only one similarity in the amino acid sequence, and then there is protein 2, protein 1 and 2, upon comparing it there is a certain similarity that you observe, and when you are comparing protein 1 and 3 you are seeing certain similarities, right? So from here you can understand that upon comparing protein 1 and 2, there is only one similarity, that is the tryptophan remains the tryptophan at position. So what I was saying is that protein 1 is similar to protein 3 more than protein 2. Why is it so? Because you see over here that in protein, between protein 1 and protein 2, you have only one residue, that's the tryptophan in position 10, that's only similar, whereas for protein 1 and 3 you have three different amino acids that are similar, like the G, S and T. So protein 3 is important. Now there are certain values that are allotted for a particular substitution. So when you're comparing the protein sequences, the scientists use the scoring systems such as the Blossom matrices. So these matrices assign scores for amino acid substitutions based on evolutionary probability. So for example, substitution between similar amino acids receive higher scores, and substitutions between very different amino acids receive lower scores. So these scores helps to determine evolutionary relationships between the proteins. So what is protein evolution? So proteins of 150 amino acids genes have 450 bases. So four types of bases, hence 4 to the power 450. That is, there can be 10 to the power 270 possible sequences. So for proteins, this becomes 20 to the power 150, that is 10 to the power 195 possible sequences. So if you start comparing this with the total number of atoms in the universe, which is 10 to the power 79 factorial, 10 to the power 79, and that's a huge number. Therefore, the probability of two randomly chosen sequences to have a significant similarity, that's 10% or more identity, is extremely low. Thus, a 20% or more sequence identity implies a common ancestor or origin. So the molecular clock of evolution suggests, so this is a hypothesis that suggests that mutations accumulate at a relatively constant rate over time. Now, this allows scientists to estimate the time of divergence between the species. So, the basic idea is genetic distance is directly proportional to the time since the divergence. However, molecular clocks are not perfectly constant. Rates may vary due to mutational rates, natural selection, population size. Therefore, modern methods use statistical models such as the maximum likelihood methods, the Bayesian approaches. Now, these methods improve the accuracy of evolutionary time estimates. So, protein evolution, that's the change in protein sequence or structure over time. So, the molecular basis is mutation, that can be point mutations, There can be insertions, deletions, gene duplication, gene fusion, and fission. So every amino acid is coded by 3 base pairs. So it's coded in the form of triplets, and each amino acid can have more than one particular codon sequence. So like for example, serine has 6 codons, whereas whereas tryptophan has only one, methionine has only one. So it's possible. Now, what are point mutations? So point mutations is a single nucleotide change in DNA. So because the genetic code is degenerate, many mutations are silent. So examples of mutations include the silent mutations. So for example, so each position in a codon can have 3 changes. Thus 9 possible mutations can happen. But for most of them, you see that you will get a glycine, right? For the other cases, you might get an arginine or a stop codon in certain cases. So what is interesting is that 9 possible mutations per codon are possible. So there can be for 61 codons, there can be 594 possible mutations. But not all of them does happen. So 25% are silent mutations, 24% are conservative mutations. There are 18% moderate like serine to alanine, 29% are drastic like glutamic acid to arginine, which is tremendous, like a negative charge becoming a positive charge. And there can be a 4% result in the stop codon. So we need to understand the different types of substitutions or mutations that can happen. So interestingly, what's important to understand is that many mutations do not significantly affect the protein function. There is a change in the amino acid, but the protein function does not change because it's not a drastic change. And there are certain proteins that have similar structures and are called the globin fold, consisting of 7 or 8 alpha helices. So sequences of several globins are aligned. Out of that around 150 amino acids, only 2 are absolutely conserved. That's possible. The histidine and phenylalanine are shown in the structure below. And the histidine side chain forms a covalent bond with the iron of the heme group. And that's conserved. So the general features of amino acid substitutions, like you have the hydrophobic residues are packed inside where the hydrophilic residues form the surface of the proteins. So side chain packaging is as dense as crystals of organic molecules. Now there are certain charged side chains that are rarely found in the interior of the proteins because they form hydrogen bonds with water, surrounding water. So also almost all interior polar groups are involved in hydrogen bonds. So together this put a lot of restraints on amino acid substitutions of residues in the interior of a protein. Hence evolutionary changes in the protein interior are much rarer than changes at the surface. So internal changes tend to compensate each other. For example, the change of isoleucine to a valine may be accompanied by an adjacent glycine to an alanine, which replaces the lost methyl group. The other factor is the insertion and the deletion, which can alter the protein sequences. So if the number of inserted bases is not in the multiple of 3, there can be a frame shift mutation occurrence. So frame shift mutations usually produce non-functional proteins. Then there is gene duplication, which adds an extra copies of the gene. So these extra copies can accumulate mutations without affecting the original function. But this process allows the evolution of new protein functions. Now homology, so proteins derived from a common ancestor are called homologous proteins. So there can be 2 types. Orthologs, so same proteins in different species with the same function, or the paralogs. That is proteins with the same organism that evolved through gene duplication. So understanding homology helps reconstruct evolutionary history. So we need an understanding of the convergent versus the divergent evolution. So divergent in evolution, so one ancestral protein evolves into multiple proteins with different functions. For example, proteases such as trypsin, chymotrypsin, elastase. And convergent evolution, where different ancestral proteins evolve similar functions. So like the chymotrypsin and the subtilin, they perform the same enzymatic function but have the different structures. Some examples are given over here. You can check them. Okay. Now we will get into the evolution of algae. But before that, if you have any questions, you can ask me. Maybe 2 minutes. If you have any questions, any doubts regarding whatever has been taught so far, you can ask. Okay, so no doubts? So I guess we can start with the evolution of eyes. So, vision evolved through multiple stages. The earliest organisms had the simplest light sensitive cells. These cells allowed organisms to detect light intensity. For example, euglena. And euglena contains an eye spot that allows phototaxis, meaning movement towards light. So the evolutionary stages of eyes are the light sensitive patches, the cup shaped eyes, you have the lens eyes, the complex image forming eyes. The anatomy of human eye, where the lens focuses light on the retina, which is composed of layers of light sensitive neurons. The primary photosensory neurons are the rod cells, which are responsible for high resolution and night vision, and the cone cells, which initiate color vision. So the rods and the cones forms the synapses with several ranks of interconnecting neurons that integrate and convey the electrical signals to the optic nerve of the brain. Now the membrane potential is reduced by the inflow of sodium and calcium through the CGMP gated channels in the outer segment plasma membrane. Now when rhodopsin absorbs the light, it triggers the degradation of the CGMP, that's the green dots, in the outer segment, causing the closure of the ion channel. So without the cation influx through this channel, the cell becomes hyperpolarized. So the outer segment is mostly filled with stacks of membranous discs containing the receptor rhodopsin, and the inner segment contains the nucleus and the other organelles. So amplification of visual signal by the rod and the cone cells, where each excited rhodopsin activates around 500 molecules of transducin, resulting in the activation of 5 PDE. So each PDE hydrolyzes around 4200 cGMP per second. So there is binding of 3 cGMP is needed to open one gated cation channel. So what is the need for the evolution of eyes? So simple light sensitive structures in primitive unicellular organisms provided survival advantages by aiding navigation and detecting light sources. Now complex imaging eyes evolved in diverse species to enhance the predation, defense and the navigation. So the light sensitive cells in primitive unicellular organisms have a primitive eye spot that helps them to detect light and move towards it. So opsins emerged in unicellular organisms over 600 million years ago, which combined with the other light sensitive molecules to enable organisms to detect light direction, darkness and intensity of light. So like the simplest light sensitive cell used for vision is the euglena. So the eye spot of euglena is red organelle made of carotenoid pigment, the granules. When light reaches the eye spot of euglena, the photoreceptor proteins present changes the shape. These changes alter the structure of flagella and influence euglena movement, and this is called phototaxis. The cup-shaped eyes are the formation. So the formation of these cup-shaped eyes provides an evolutionarily significant event where the flat light-sensitive cells evolved into better curved structures. Now this shallow depression allows the light-sensitive cells to detect the direction of light. Now light reaches the cells through an angle, and as the depression deepens, the direction of light becomes finer. Flatworms have cup-shaped eye. The cup-shaped eye detects changes in light intensity, helping in navigation and detecting the predators. Now the formation of the cup-shaped eyes provides various advantages, such as the ability to detect the direction of light, sense environmental changes and enhance the survival of an organism. So the cup-shaped eyes do not form images as they are only able to detect light and dark. Now in the process of evolution of eyes, the development of cup-shaped eyes plays a crucial step towards more complex eyes, indicating important changes and adaptation over time. The lens size, it's found in vertebrates such as the humans. So they have advantages of high resolution and depth perception. So lenses collect light and bend precisely onto the retina for image formation. This helps to collect the fine details of the objects and understand the surrounding in a three-dimensional manner. Some unique adaptations of lens in eyes. So fish, they can analyze changes in the refractive index underwater. Eagles have extremely sharp vision. You have the cephalopods that have high adaptability to low light underwater conditions. The chameleons that have independently moving eyes provide a broad field of view for spotting prey, predators, and humans that can differentiate between different colors and have high resolution vision. The compound eyes are mostly found in insects. They have advantages like wide field of vision, excellent motion detection. So compound eyes are composed of multiple repeating units called the omatidia. So each of these functions as an individual visual unit having its lens and photoreceptors. So rapid motion detection, adaptability becomes quite important, those who have compound eyes. So bees have compound eyes that can detect UV patterns on flowers and help them in for pollination and navigation. So complex eyes are advanced visual systems composed of different structures that can provide high resolution images. So these structures include lenses, retinas, and the photoreceptor cells. So complex eyes have advantages like I mentioned, high resolution images, advanced light sensitivity, color vision, quick motion detection, things like that. There are certain protective membranes and shields that safeguard the different outer portion of the eye from the environmental damage, infections, physical injury. So that is why you have the eyelids present in humans that protect you from dust, debris, and foreign particles. Tears have enzymes, lysozymes that fight microbes and keep them moist and healthy. And the nictitating membrane, that's the third eyelid in some animals like the birds and the amphibians, that provides extra protection without obstructing vision. This is just a table of the advantages and the disadvantages of the different types of eye. You can find also the difference between an octopus versus a human eye. I'm not going into all of these details. What's more important is that model organisms are species used to study biological processes. They are chosen to grow because they are easy to grow, genetically traceable, representative of broader biological systems. So models organisms have characteristics like a rapid life cycle and reproduction, genetic similarity to humans, and the availability of complete genome sequences and the genetic tools. Cells are used as model systems in research because they're easy to manipulate. Using cells instead of the whole organisms reduces the ethical concerns. It also allows high throughput studies like if you want to undergo drug screening or molecular interactions, and for in vitro cell studies it allows the researchers to focus on the specific biological mechanisms without the interfering into the complexity of an entire organism. So like you have the HEC293 cells, that's derived from human embryonic kidney cells. They have advantages of high transfection efficiency, widely used for protein expression. And disadvantages like they are not fully represent the specific tissues or primary cells, which will obviously remain because you are not using the entire system or the entire organism. Then you have the HeLa cells that are derived from the cervical cancer cells. So they have advantages of uh, robust growth, widely used in cancer and virology research. And the advantages is that as they're cancerous, so uh, results from the HeLa cells may not always represent the normal cell behavior. Now, prokaryotic models, you have the E. coli. You have heard of it. The advantages is that it has a rapid growth. It's used for different biotechnological purposes. You can do recombinant technologies, CRISPR system. You can incorporate the CRISPR systems. Uh, do all sorts of experiments on this because they have easy genetic manipulation, widely used for cloning and protein production. So there are different advantages. It's a wide advantages of E. coli. That is why it's widely used a prokaryotic system in research. Disadvantage is that it lacks organelles and eukaryotic specific processes. So it's only a bacterial system or a prokaryotic system. You cannot use this data for a eukaryotic system. Then prokaryotes have an operon model. So operon model is a fundamental concept in biology that explains how genes are regulated in the prokaryotes. Now the unicellular eukaryotes as model organisms like the Saccharomyces cerevisiae, it possesses many of the same cellular structures in multicellular eukaryotes like the nucleus, mitochondria, endoplasmic reticulum, Golgi apparatus. So many key cellular mechanisms such as cell cycle, DNA repair, transcription, translation occurs. Many unicellular eukaryotes have well characterized genomes and are easily manipulated using the genetic tools. So yeast system is mostly used for studying cell cycle, metabolism, gene regulation, and you use it for the yeast 2 hybrid system that allows the researchers to study protein-protein interactions. So animals and model organisms like you have the fruit fly, the Drosophila melanogaster, the nematodes, that's the uh, C. elegans, you have mouse, that's musculus, and these organisms helps you to study the development, genetics, and the disease mechanisms. So mainly the fruit fly is used because they have shorter life cycle with high reproductive rates, and the clutch size, the Drosophila genome is small and fully sequenced, so you can use extensive genetic tools such as RNA interference, CRISPR-Cas9 if you want to do it. And also many biological pathways and genes are conserved in Drosophila, that's same as the humans. Right, so homeotic transformation occurs when one body part is replaced by another due to mutations, like you have heard of the homeotic genes or the Hox genes. Now these genes play a central role in developing the body plan of an organism. So homeotic transformation occurs when one body part is replaced by another due to mutation. Now these genes play a role in the body plan. So what happens if the gene gets mutated or suppressed? What you will see that in the position of the leg gets exchanged, the position of the wings gets exchanged, and things like that happen. The C. elegans is used because they are small, consists of 1,000 somatic cells, has a fully developed nervous system, digestive system, and a reproductive system. Its transparency allows researchers to observe cellular and developmental processes. And it's the first multicellular organism that was studied for which the entire cell lineage was mapped. And the discovery of key genes involved in apoptosis provided insights into the genetic control of the cell death. So RNA interference was mostly demonstrated by Andrew Fire and Craig Mello. So injecting double-stranded RNA into the C. elegans silence specific genes more effectively than the single-stranded RNA. So this effect was heritable as subsequent generations of the worms also exhibited silencing of the target gene. And mouse model or the is one of the most widely used model organisms in biomedical research. So 99% of mouse genes have homologs in humans. Mouse models mimic human diseases such as cancer, diabetes, cardiovascular diseases, neurodegenerative disorders, Alzheimer's, Parkinson's, infectious diseases. Mice is used for preclinical testing of drugs to study the toxicological effects of various compounds. It's used for studying memory, learning, anxiety, and the social behaviors. So induced pluripotent stem cells were first generated by Shinya Yamanaka and Kazuchika Takahashi in adult mouse fibroblasts. So they hypothesized that genes important to the embryonic Stem cell function might be able to induce an embryonic stage in adult cells. Now, plants, what do you mostly use is the Arabidopsis thaliana, which is used extensively in the plant genetics. The tobacco is another which is important for studying plant virology and the gene expression. So, Arabidopsis is mostly used in plant biology. So, they are small and fully sequenced genome. They have high efficiency of genetic transformation. They completes the life cycle in around 6 weeks. So, rapid growth makes them suitable for uh, experiments requiring multiple generations. And tobacco, so it has a high transformation efficiency and a stable integration of foreign DNA into its genome. Tobacco, due to its large leaves, is a leading system for chloroplast transformation, enabling high levels of foreign protein expression. The tobacco mosaic virus have been the vital tool for studying viral infections and plant immune responses using tobacco as a model organism. And the tobacco mosaic virus was the first virus to be discovered. The infection causes characteristic patterns such as the mosaic-like mottling, and discoloration on the leaves of tobacco. In 1892, Dmitry Ivanskyi first gave the concrete evidence for the existence of non-bacterial infectious agent, showing that the infected sap extracted from the TMV tobacco leaves remain infectious even after filtering from the finest Chamberlain filters. So, to summarize, life on Earth evolved through a series of major transitions: origin of life, oxygenation of the atmosphere, emergence of eukaryotic cells, evolution of multicellular organisms, diversification of animals. Now, molecular biology has greatly enhanced our ability to study evolution through sequence comparisons, molecular clocks, protein evolution studies. So, the model organisms remains the essential tools for understanding the biological complexity and is widely used in different research nowadays. Okay, so with that, we can move on to the quiz part. Okay, so in the meantime, if you have any questions, you can ask me, maybe 2 minutes to that. If you have any questions, you can put it up in the chat box. Okay, so if there are no doubts, we can start with the question one by one. You can give me an answer in the chat box if you want. So the first question says gene duplication plays a major role in protein evolution because. So the first question, gene duplication plays a key role in protein evolution because. What will be the answer? So what does gene duplication do? It creates redundancy. So when an organism has two copies of a gene, one copy can continue to form its essential biological function, ensuring the survival. So this relieves the selective pressure on the second copy, allowing it to accumulate mutations freely. So the correct option is A. It allows one copy to mutate and acquire new functions while the other retains the original function. The concept of molecular clock in protein evolution is based on. So the molecular clock hypothesis, what does it suggest? That for a given protein or DNA sequence, mutations accumulate at an approximately constant rate over the geological time. Now, because this rate is relatively steady, scientists can compare the difference between the sequence of the two species and estimate how long they diverge from a common ancestor. So the correct answer is the regular accumulation of mutations over time in a protein sequence. That is option B. Now the next one. What is an orthologous protein? What is an orthologous protein? So by definition, orthologs are types of homologous genes or proteins that are found in different species but share a common ancestor through a speciation event. So the correct answer is C. Option A describes the paralogs which arise from gene duplication within the same species. B and D do not accurately define the evolutionary relationships of orthology, which is specifically about the shared ancestry across the species. The next is what is the primary reason for using model organisms in biological research? Quite easy, quite simple. Why do we use model organisms in biological research? We use them because they are simple, easy to maintain, they provide insights into the basic biological processes. Some have shorter life cycles and some are inexpensive, some are better, some represent quite, they have great similarities with the human beings. So that is why it gives us a understanding of the bigger picture, even though they are model organisms. So option B. Okay. The next one is on Drosophila melanogaster. So it's considered a valuable model organism. Why? So why is Drosophila considered as a valuable model organism? So if you go back and look into the advantages of Drosophila melanogaster, you will understand that they have what advantages? Rapid reproduction, genetic simplicity, and are obviously cost effective. So they have a very short life cycle, about 10 to 14 days. So scientists can study many generations in a short time. They have a relatively simple genome with only 4 pairs of chromosomes, yet many of their genes are similar to the human genes. And they are small, inexpensive to maintain, and easy to manipulate genetically in laboratory setting. So that is option B. Okay, now something quite interesting. So what is the Cambrian explosion and how is it related to the evolution of the eye? So how do you link the Cambrian explosion to the evolution of vision? So the Cambrian explosion, it refers to a period that was long, million years ago, where most of the major animal phyla first appeared in the fossil record. It wasn't a literal explosion, but a relatively sudden burst of evolution diversity. Now, this period saw the first appearance of complex image forming eyes, like the compound eyes of trilobites. So, the researchers, the scientists believe the evolution of vision may have been a primary trigger for the explosion, as it intensified the predator prey relationships and drove the animals to develop new survival strategies like shells and faster swimming. So the correct answer is B. That's the rapid diversification of life forms, which included evolution of complex size in several animal groups. Okay, another question on vision. So which of the following represents a key step in the evolution of the eye from a simple light sensitive spot to a complex camera like eye? What will be your answer? Yes. So the development of a lens to focus light, the appearance of a retina with photoreceptor cells, the formation of a transparent cornea. So all of these points are the important steps in the evolution of eye. That is option D. The next one says, uh, what key event in Earth's history led to evolution of aerobic respiration in organisms? No, that's not the Cambrian explosion. So early Earth had an atmosphere with almost no oxygen. So cyanobacteria began forming oxygenic photosynthesis, releasing oxygen as a byproduct. So the great oxidation event filled the atmosphere with oxygen. That is the rise of oxygen in the atmosphere due to photosynthetic organisms. That will be correct. That is option B. And the next two, in multicellular organisms, the ability of cells to adhere to one another is typically facilitated by the presence of. So the cells adhering to one another is typically facilitated by the presence of the cell adhesion molecules. So these are specialized proteins located on the cell surface. They allow the cells to bind with other cells or with the extracellular matrix. Now, this process is critical for maintaining the structural integrity of the tissues and for cell-cell communication. Okay. Now, one question from protein part or an amino acid part, whatever you say. Which of the following pair of amino acid substitution is most unlikely to happen? Leucine to aspartic acid, threonine to serine, phenylalanine to tryptophan, lysine to arginine. So which of them cannot, or it's very difficult to happen? What will be your answer? Let's see from lysine to arginine. Both are positively charged, that is basic amino acids, and often function similarly in the protein structures. The phenylalanine to tryptophan, both are large aromatic non-polar amino acids. That is also possible. D is definitely possible. Threonine to serine, both are polar, contain a hydroxyl group. So they are very similar in size. And leucine to aspartic acid. So leucine is non-polar, hydrophobic, while aspartic acid is negatively charged, hydrophilic. So changing a water-fearing amino acid inside a protein to a charged one would likely to destabilize the protein's entire structure, and that is most unlikely to happen. So in evolutionary biology or protein chemistry, substitutions are likely if two amino acids share the similar chemical properties like size, charge, hydrophobicity, and that is called conservative substitution. Fine. So option A is most unlikely to happen. Okay. Yeah. That's all from my part. If you have any doubts, you can ask me. I'll be sharing the slides and the video will be uploaded in YouTube. You can check from there if you have any questions. So thank you very much for joining. We will again meet in the next week on Friday at 6 o'clock. Yeah. Thank you and yeah, have a good day.